So now I'm going to talk about DNA. And I'm going to start with the makeup of DNA. You know, what are these chemicals that make up deoxyribonucleic acid? So that's what DNA stands for, deoxyribonucleic acid, as it says at the top. And its structure, you've probably heard about, is a double helix. A double helix looks like a twisted rope ladder. And the rungs of the ladder, where you would put your feet if you were climbing up, okay? The rungs of the ladder are these nitrogenous bases. And there are 4 of them. Bases are the chemical opposite of acids. You remember that from chemistry class a million years ago? Ammonia versus Clorox kind of thing. Okay? Don't put those 2 together. It'll blow up in your face and you can die. But those four nitrogenous bases that make up your DNA are adenine, thymine, cytosine, and guanine. There is going to come a time when another one comes into play called uracil, but I'll come back to that. The edges of the ladder, the sides, like where you would put your hands if you were climbing up, those are sugar phosphate molecules, or phosphate sugar molecules. A phosphate and a sugar put together to make a molecule, and they are for structural support. They are the backbone that holds the DNA, the nitrogenous bases, in that order. The bases always pair up the same way. Adenine always pairs with thymine, and cytosine always pairs with guanine. A always goes with T, and C always goes with G. Every rung has 2 bases. And if one of the sides is the G, the other side will always be the C. And if one side is the A, the other side will always be T, and so on and so forth down the whole length of the DNA rope ladder, the DNA double helix. And while there are only 4 possible bases, 4 types of bases, there are like 3 billion total in the genome. So a huge number. And that genome is the code for making you. Yes?
3 billion pairs of the...
Oh gosh, I don't know. I don't know if it's 3 billion individual, 3 billion pairs. I'd have to look it up. Good question. I think it's pairs. So the genetic code does a number of different things. Number 1, you can reproduce the whole genetic code, make a complete copy of every single part of it, and that's what mitosis is. Mitosis is making a whole new cell, a whole new body cell, a whole new cell that is you. Meiosis is making a gamete, a kind of half cell. But the other thing, and so that gets half, each daughter cell gets half your DNA. But the third thing, these are both kinds of cell replication. The other thing that DNA does is protein synthesis. Because early on, you are this undifferentiated stem cell. And then you start to make more of those, and you're still just a clump of undifferentiated stem cells. There's no functional part of you in there. When they differentiate, then they become the things that they're going to be. And so the neuron, if you've been following along, every neuron is like a city of activity. It has a nucleus that is filled with DNA and all kinds of organelles. It has dendrites that have all different kinds of receptors and reuptake channels and second messenger systems and all of that good stuff. Microtubules. So there are all these different parts of the neuron. In the end feet, then you're going to have, the vesicles, and they're filled with neurotransmitter. So every cell has lots of things going on, has lots of pieces. And those pieces are built according to the instructions in your DNA. So, let's pretend like someone in this class is learning something, okay? Let's just pretend, okay? I know, right? It's weird, but we'll just pretend it for a second, okay? So let's pretend that someone in this class learned something, and they need to build a new AMPA receptor. Remember that, LTP? Oh my God, somebody learned something, okay. People remember LTP, okay? A message needs to go to the DNA that says, make a new AMPA receptor. So the DNA has a code for building an AMPA receptor. An AMPA receptor is built, I don't know what the codes are. I don't have the actual genetic code memorized. That would be crazy, okay? But you have a code. And you read that genetic code, and if you put those substances together that way, it makes the protein that is an AMPA receptor. So you'll have a thing here that says start here for making an AMPA receptor and stop here for making an AMPA receptor. And in between, put it together like this. So your DNA is a set of instructions for making all of those pieces of you. That's why there's so much of it. Because not only do you have to code for every receptor on every neuron, you have to code for everything that's in a blood cell, or a heart cell, or a lung cell, all of the cells. You have to have all of that information in the DNA. And so that's why it's very long. Every set of 3 nitrogenous bases is called a codon. And the reason it's called a codon is because if you put those 3 bases together like that, it codes for an amino acid. So this is a code for an amino acid, and this is a code for another amino acid, and this is a code for another amino acid, and this is a code for another amino acid. And when you put together all those amino acids, that is a protein. Amino acids are the building blocks of proteins. So the bases, the base pairs, are the instructions for putting together an amino acid, and then you link together all of those amino acids. That's a protein. So this is one gene. From the beginning of where you start to make the protein to the end of where you stop making that protein, that's a gene. A protein recipe. And a gene is made out of sets of 3. Those are codons. So again, I remind you that DNA has two functions, but one of those functions can be broken down into two places. Replication can either be mitosis or meiosis, okay? Both of those things count as replication, making a new cell. But protein synthesis is where you build just that one little piece, just that one little protein. And that happens in steps. In the first step, we unzip the DNA and we start reading. And another bunch, another set of bases is going to come in and match up to the DNA. But remember that the way they bond is the opposite likes to hook up with it. So if there's a G on one side, it's going to be a C on the piece that's being copied. This piece is called RNA. When it's just the one side, it's RNA. It has to have the two sides to be DNA. But RNA comes in and makes a mirror image of the one side of the DNA. Okay, it makes a mirror image. So, this is where uracil comes into place. Remember I said uracil? Uracil takes the place of thymine during protein synthesis. Uracil takes the place of thymine during protein synthesis. During transcription, this RNA comes in and creates the mirror image of the DNA. The RNA here is called the messenger RNA because it's not the final product. It's the mirror image of the final product. In the next step, translation comes in and re-reverses it. Translation comes in, and now you have what was cytosine back over there being flipped back into guanine. And so now you are assembling not the mirror image, but the original image, right? You're flipping it back. At that point, the RNA over here that flips back, that does mirroring, the re-mirroring, the second, flipping back to the way it originally was, that's called transfer RNA, tRNA. And so as the codons go down and this says, hey, make this amino acid, and this says make this amino acid, and this says make this amino acid, and this says make this amino acid, you build all the amino acids, you link them together, and that's your protein. Yes?
So we need to transfer it back so we can understand what the heck that we just coded?
Okay, so like let's say that I wanted to send a message to you and I could only communicate in pluses and minuses, and my double helix looked like this, okay? They always have to bond positives to negatives, right? Okay? So if I want to make something that looks like this, the first step is to unzip and make the mirror image of this, because a positive would repel another positive. If it was two thymines, they don't go together, they don't match up. Okay? So you have to work with what you have, the forces of nature that you have. Which is that first we can build the flip, the reverse, and then we can reverse it back to the original structure. Okay. So, all of this happens in the nucleus of the cell where that DNA is, and the surrounding organelles help with this process. So ribosomes, for example, are the site of protein synthesis. The ribosomes hold the DNA in place, the RNA in place, so that when the tRNA comes in, it can match up with it and form that structure. Any any segment of DNA that codes for a protein is called an exon. Because the protein gets built and then it exits the nucleus. Okay? So the protein gets made in the nucleus, but then it gets sent to wherever it needs to go. Okay, I made an AMPA receptor. Now you have to go where you're supposed to go. Okay, I made a serotonin transporter. Okay, now you need to go where you need to go. So those are called exons. Anything that codes for a protein that leaves the nucleus is called an exon. But there are other parts of the DNA that don't code for proteins. And those are called introns. Because what they do never leaves the nucleus. It stays in the nucleus. And we know some of the things that these introns do is to regulate the function of the exons, is to regulate the function of the proteins. So for example, not only are there all the codons that code for the protein, there's also a codon at the beginning called a start codon and a codon at the end called a stop codon that say, here is where you start making this and here is where you stop making it. We used to think that all of the places on the DNA that didn't code for proteins, we just called them junk DNA. And we thought, oh, well, they're there just to like absorb random mutations so that, random mutations don't affect us so much. No, it turns out that they regulate the proteins. They regulate the things that are going to leave the cell. Okay. However, there are other functions that continue to remain unclear, and we are learning more about them as time goes on. Yeah.
So start and stop would be the introns then, and everything else.
Anything that makes a protein is an exon. Start and stop codons are example of introns, but there can be other things that do other kinds of regulation or other things as well that aren't just start and stop codons. That's just one example of an intron. Does that make sense?
Yeah.
Okay, cool. All right. I'm going to stop and take a break right there. Okay, so you can have changes occur to the structure of the DNA, and we call those mutations. And they can occur in a number of different ways. Point mutations, insertions, deletions, frame shifts. And I'm going to talk about what those are right now. You can think of a gene as a sentence made up of a bunch of three-letter words. So, you can have a start codon right here that says start reading the DNA right here to make the protein, or start reading the sentence right here, okay? And let's say, the codons go something like this. I'm just making up anything, okay? Like I say, I don't know what the different codons are, okay? And then you have stop codon. It's kind of like if you have a sentence that says, the boy ate the, well, let's say there's one more word. Ate the, and these thes would be the same, right? The boy ate the cat. weird boy, right? Okay. Okay. So let's say it's a sentence like that. Now, you may notice that this codon is a little tiny bit different than this codon, but they still make the same word. Sometimes an amino acid can be built two slightly different ways. And there's a list of them in your book. So they'll give you a list of the different amino acids and all the different ways you can build those amino acids. Sometimes there's just one way to build it, but sometimes there's a couple. But let's say that instead of a G right here, there was another C. And instead of it being the boy read the cat, or the boy ate the cat, it changes it into the box ate the cat. Now you have a different sentence. That kind of change is called a point mutation. And it's a point mutation where you have one single base that changed that led to creating a new amino acid here, or like a new word in this sentence. Does this make sense, the analogy I'm giving? Yes.
So it's possible for two amino acids to be built two different ways, but it's a point mutation if it creates a whole new amino acid?
So this and this, probably the way that there's a difference is that they were caused by a point mutation. But in this circumstance, it builds the same amino acid. It still builds the word the. It still builds, I don't know, whatever, lysine or some amino acid, I don't know. Like I say, I don't have the chart of amino acids memorized. I'm sure there are people who do. I'm not one of them. But it ends up building the same amino acid, so it doesn't really matter in terms of the functionality of the protein. But sometimes the difference does matter, and it changes the functionality of the protein. And so that's the kind of thing that we mean by different alleles. You changed something that mattered to the outside expression. You changed the genotype and it affected the phenotype. That's how we get different alleles. That's how brown eyes, one time there was some kind of mutation, and then there was a blue-eyed child. Sometimes, though, you can get other things. So for instance, you can get an insertion. And sometimes you insert a whole new codon, okay? So let's go back to the way it was before, okay? The boy ate the cat, and you can insert a whole new codon, and let's say it reads, like the word red. So now you have the boy ate the red cat. So now you have a protein that has one extra amino acid in it. That's an insertion. Or you can have a deletion. And it's possible that the deletion takes out a whole word. So now it just says the boy ate cat. It took out a whole codon. The boy ate cat. You're missing one of the amino acids you had before. But sometimes it doesn't happen in sets of 3. Sometimes something gets inserted or deleted and it's just one base or 2 bases. And so this is what happens. Let's say that you have TAA, CCG, GCT, and it reads the boy ate the cat. It makes this protein. Let's say this base got deleted. Just that one base. Well, codons get read in sets of 3. So when you start making this protein, the first amino acid built is going to be this. Then the second one is going to be this. Then the third one is going to be this. In other words, it shifts it all the way down. From the point where the mutation is either inserted or deleted, everything from that point on is going to be changed. That's called a frame shift. Now you're making totally different sets of amino acids all the way down, and so you have a completely different protein. So a frame shift, especially if it occurs early on in the genetic code, has enormous impact on making that protein. It can make something that's completely non-functional. Does that make sense? Okay. Sickle cell anemia is an example of a point mutation. You have one difference. Instead of there being an A, there's a T. And in this case, it makes a completely different amino acid. And so you have different alleles. And if you have one of them, you will have sickle-shaped blood cells. And if you have the other, you'll have round-shaped blood cells. Early onset Alzheimer's disease is an example of a single gene having a mutation and being responsible for the disorder. So if you have the gene for early onset Alzheimer's disease, we know that it's this one gene that's affected that creates that disease. However, for late onset Alzheimer's, for the one that progresses more slowly and occurs in your later life, the one that occurs in like your 70s and 80s, typically, that kind of Alzheimer's disease, a whole bunch of different genes contribute to it. So with the early onset Alzheimer's, it's kind of like you either have the gene for it or you don't. With late onset Alzheimer's, there are a number of different genes, and depending on which alleles you have, it can make you more susceptible to Alzheimer's or less. There are actually some alleles that seem to offer some protection against late onset or typical onset Alzheimer's. The most common one neither increases nor decreases your risk, and then there's another one that increases your risk. Cystic fibrosis is a disorder of the lungs, and the lungs are incredibly complicated. And in fact, you can have one of any of 1,000 different mutations, and any of them can cause you to have cystic fibrosis. The lungs need to be built in a very specific way, and there's a lot of functional parts, and if any of them gets a slight mutation, it can result in cystic fibrosis. Huntington's disease, I don't want to go there yet. Huntington's disease is usually produced by something called a triplet repeat. What that means is one of the codons keeps getting repeated. So it would be like a sentence that said, the boy ate, the cat. And And what it does is when it has a whole bunch of those amino acids in the middle, or anywhere in it, it builds a big junky protein that doesn't work. And the protein, the junky protein that it happens to build isn't useful, but it builds up in your brain. And so over time, it kind of squishes out the rest of the brain. And so as this protein accumulates in your brain, it kills you. Huntington's disease is caused by a dominant allele. So if you have one allele from one of your parents that's for Huntington's disease, you will also get Huntington's disease. But the reason that they can pass that on to you, that they can pass on a dominant bad gene, is because it doesn't kill you until your adulthood. People can have Huntington's disease and be making the proteins in their brain and have no idea it's happening. And then in their adulthood, when they start to experience symptoms, then they get diagnosed. And they may have already passed that on to a child. Now, if they had one unaffected gene, then they could 50% of the time pass that good one on, but 50% of the time they'll pass on their bad one, and then that kid is going to get Huntington's disease as long as they live long enough. I mean, it's always possible you get hit by a bus when you're 23, and then it would never manifest itself. But that's the way Huntington's disease is usually caused. Occasionally, you get a thing where the stop codon doesn't work, and it just keeps going and keeps reading the genetic code for too long. But again, the same thing. You just have a bunch of junky amino acids making a big old protein that doesn't work. But most Huntington's disease comes from the triplet repeats. But it's not all bad. Most of the time, when a mutation occurs, it's not a good thing. Most of the time, if you have a mutation occur, it results in things like cystic fibrosis, Tay-Sachs disease, what have you. But sickle cell anemia is a good example of a mutation that does serve a purpose. There's a reason that it has survived in our genes, in some of our genes. And that's because having sickle-shaped blood cells protects you against malaria. Malaria cannot kill a person who has the sickle-shaped blood cells. And so if you just got a mutation that happened to give you the sickle-shaped blood cells and you lived in an area that had a lot of malaria, you actually would be more likely to survive. Yeah, your blood's a little bit junky, but at least you didn't die of malaria, you know? So that's why certain populations, for example in America, who came from, their origins are from places that had high rates of malaria, they will have higher rates of sickle cell anemia. So, in other words, mutations create new alleles. And sometimes it has a beneficial effect. Most of the time it doesn't. Sometimes it's neutral. So for example, the allele that evolved for blue eyes. Okay, well you have blue eyes. It's different, but it doesn't help you or hurt you. So, some people have blue eyes and some people have brown eyes. Doesn't matter. Persists in the population because nothing's selecting against it. So, you'll just see it come up from time to time. But every once in a while, you'll have a mutation occur that creates something in the offspring that is beneficial. So, for example, let's say I'm a little deer that lives in the forest. And I'm a little brown deer and all the other deer are brown. But I have a mutation in one of my eggs that makes that egg have code for striped fur. And now I have a baby that's born with striped fur, and it just so happens that that hides it even better in the forest. Well, if that baby can hide even better, it's more likely to survive and reproduce. That's how evolution occurs. Evolution occurs by random mutation, and then nature selects, natural selection, nature selects which ones work versus which ones don't work. And that process happens slowly and randomly. The mutation process happens every once in a while and then gets selected for or against. And if it gets selected for, then we have speciation that can occur. We can get new species if the changes are big enough. And if something that was previously an advantage, now the environment changes and it becomes disadvantage, it can be selected against. So let's say you're the animal that has all the little stripes, and now you're not in the forest anymore. Now it's, the weather changes and it's snow everywhere. Well, now you might really stand out, and so now maybe you'll get eaten. So that's why species can change back and forth in different ways in different places. All cells can mutate. And we call any mutation of your body cells, okay? The body cells that make up you, if they get a mutation, we call that a somatic mutation. It's just affecting you, your somatic cells, the ones that make you. So for example, skin cancer. You have, all of your skin cells chilling in your arms, and light rays hit them, the sun rays hit them, you go out in the sun, and it mutates the skin cells. And if it mutates one, and it starts to just reproduce like crazy, that's what skin cancer is. But that doesn't affect your eggs or your sperm. It just affects you. If your eggs or your sperm are the one that undergo a mutation, we call that a germline mutation. And those are the ones that result in an offspring that's different than the parent. And in fact, that wouldn't affect you as a parent. If there was an error in replication in making your sperm or your egg, you wouldn't know. But your offspring might manifest that change. Your offspring, if they survive, might show that difference. Any gene that has more than one allele is called a polymorphic gene. Poly, many, morphic, forms. So, blue eyes versus brown eyes, versus green eyes versus purple eyes, polymorphic. There are many different alleles. Whereas the one that codes for instance, having 5 fingers on your hand, there's just that one allele that we all have. But that's not the only thing that can affect how DNA is expressed. There's another thing called epigenetics that we're learning about. And epigenetics, epi means above or around genetics. And what happens in epigenetics is you change which of your genes is being expressed. So there are different points in your life when different genes need to be expressed. So for example, when you go through puberty, you have genes that code for things like armpit hair and pubic hair, but they're turned off before you go through puberty. And then they turn on when you go through puberty. You also have genes within you that code for making a placenta and making breast milk. But unless you're pregnant or having a baby, or having had a baby, those genes are not expressing themselves. If you're under a lot of stress, you can turn on certain genes and turn off other genes. So in other words, you're regulating those regulatory processes. Which genes are expressed and which genes are not. Okay, now I'm turning up how much AMPA receptors we're making, or now I'm turning down how much AMPA receptors we're making. And here's the thing, let's say, so for example, you're a woman and you're under a lot of stress, and so you get the epigenetic tag for making stress hormones like cortisol, okay? So you're making a lot of cortisol, and so those genes are active. If you are pregnant, you can also activate those genes in your kid. So your kid is in there, they're not the one under direct stress, you are. But by turning on your genes for those stress hormones, you can turn on their genes too for making the stress hormones. And if it's a female fetus, at a certain point in development, she has all her eggs. So not only can you turn on the genes in the female fetus, you can turn them on in her eggs too. And therefore affect gene expression all the way down into your grandchildren. Now you're not changing the genetic code. The genetic code stays the same. You're just turning on or off which genes are being expressed.